Welcome to another lecture of Developmental Biology lecture series. This lecture is also about teratogenesis. In an earlier lecture, we have studied that certain chemicals that are called endocrine disruptors, they have potential to cause tumors, or they can, in other words we can say, predispose animals with the risk of cancer. We had studied that when a fetus, or what you can say, when a human being is exposed during its prenatal life or its fetal life to these endocrine disruptors, some of these endocrine disruptors, that human being, when he's or she is going to become an adult, there are going to be chances that he or she is going to get some kind of cancer. Especially we studied about the breast cancer in females who are exposed prenatal to endocrine disruptors. So, with the passage of time and with more and more research being done into this topic, we are now increasingly interested in studying cancer as a disease of development, and this is what we are going to do today. So, though it is a kind of a, what you can say, a novel approach, but it is not a novel approach at all. The idea has been here since long. For example, it says that it was first proposed in 1953 and then 1961 and then 1978 that there might be some connection between what you can say oncogenesis and teratogenesis. Now, carcinogenesis is more than just the genetic changes. Normally, we think of cancer as only the genetic changes, the mutations that are going to give rise to tumors, but it is more than that. Rather, carcinogenesis, or what you can say oncogenesis, it has been viewed as aberrations of the very processes that underlie differentiation and morphogenesis. Means that it is not just the genetic makeup, the changes, the alterations, the mutations in genes that may give rise to tumors, but also the processes that are behind differentiation and morphogenesis during the embryonic life, during the prenatal life. These changes or these abnormalities in such processes such as differentiation and morphogenesis, they can also lead to cancer. And in a very recent study, you can say in 2016, this study was of the melanomas, which are cancers in zebrafish. This study demonstrated that when melanocytes or pigment cells had two of mutations that are found in human cancer cells, they formed pigmented nodules, but not tumors. So these melanocytes, with two mutations that are found in numerous cancer cells, they were not becoming tumors. They were supposed to become tumors, but they were not becoming tumors. Rather, they were becoming pigmented nodules. Now, these melanomas, these are cancerous growth of melanocytes, or what you can say, the cells that contain melanin and give us the pigmentation that we have. So these melanomas, or the cancerous growths, or in simple word, the cancer of melanocytes, it occurred only when the pigmented cells also expressed markers of neural crest precursor cells that had given rise to melanocyte in embryo. So let me clarify this condition a bit. So what happens is that though the mutations are present, yet these melanocytes, they are not going to form uh, tumors or cancers, that they are forming pigmented nodules. However, when are the cancers formed? Then when these melanocytes become malignant melanomas, well, that happens when these melanocytes, they start expressing the markers of neural crest precursor cells, or what we can say, when developmental abnormalities, they come along with the mutations. So these neural crest precursor cells, their markers, they were supposed to be present during the embryonic life, but not in the adult life. And when they have somehow re-originated, or somehow you can say their expression has resumed, that is going to cause melanomas, or the cancer growth of melanocytes. And thus, teratogenesis, the developmental disorders, are somehow linked to cancers. Now, in the early days of research of cancer research, it was thought that carcinogenesis, or the formation of cancer and metastasis, what you can say, the spreading of cancer to other tissues and etc. in simple words. So it was thought that this carcinogenesis and metastasis they were caused by but the proliferation of a cell that had acquired mutations that enable it to become autonomous. Or in simple words, we can say that earlier it was thought that cancer is just an uncontrollably dividing cell. Thus, defining cancer by intracellular mechanisms that enable cell to become independent of its environment. Or in other words, we can say that it was thought that only uh, the inside of cell, only the genes that are present in cell, the genetic makeup of cell, that is going to get involved in oncogenesis or what you can say in carcinogenesis and metastasis. And there is no role of nearby cells or the environment of cell. But this turns out to be only a part of explanation, or in other words, this is not the complete story. Now we have realized that the initial cancer cells they modify their environment, and they modify their environment. How they modify their environment? They modify their environment into some cancer promoting niche. Or in other ways, we can say that cancer cells do require some environmental changes in order to be able to undergo metastasis. So what cancer cells do, the initial cancer cells, they modify the nearby environment or what you can say the nearby cells and tissues and organs so that they can grow freely or their growth is promoted by such environment. So cancer is being recast as a result of a stepwise progression of conditions that depend on reciprocal interactions between the incipient cancer cells and supporting cells of their tissues. Simpler words, we are trying to say that we are now trying to study cancer not only as a genetic problem but also as something that is related to the reciprocal interaction between the incipient cancer cells and between the supporting cell of their tissue environment. Or in other words, we can say that cancer cells they are influencing surrounding tissues, they are influencing their environment, and then reciprocally their environment which they're modifying, that environment is supporting that cancer's growth. Now, this progressive alteration of what you can say cancer cell interactions, it leads to aberrant tissue architecture, or in other words, what we can say that the whole tissue is modified or it is deformed or it is undergoing abnormal changes just because the cancer cells are modifying it in order to able to uh, what you can say grow more and more, and possibly formation of niches that generate cancer cells. That tissue is now going to what you can say build niches that are going to promote the generation of more and more cancer cells. So indeed, cancer cells appear to proceed by recapitulating steps of normal development, or what we can say that whatever happens during normal development, the cancer cells are just going to uh, they are somehow able to what you can say make that happen again, and they are going to make those normal developmental changes resume again during adult life. uh, those changes that were during embryonic life, these cancer cells are somehow making those changes to occur again during adult life in order to make these cancer cells able to grow. So they include the formation of a niche which to proliferate. Thus, both the carcinogenesis and the congenital anomalies, these can be seen as disease of organization, differentiation, intracellular communication. Thus, we can say that cancer has a close link with developmental biology, or we can view cancer as a disease of development. Now, we are going to see that there are these cancer, they are often caused by defects in the same pathways. Uh, these pathways that are going to what you can say mediate or control this organization or morphogenesis and differentiation and intercellular. Now, there are many reasons to view malignancy as metastasis form of development, and four of which are discussed uh, four of which we are going to discuss as a part of this textbook, Developmental Biology, 11th edition. Now, the first type is a context-dependent tumor formation. The second one is defect in cell communication, and it can initiate cancer as well. And then is cancer stem cells, and then there may be epigenetic reprogramming of cancer cells. Now, let's get to our first subtopic, the context-dependent tumors. So, what are context-dependent tumors? Well, many tumor cells they have normal genomes. Many tumor cells they have normal genomes, and whether or not these tumors become malignant, it depends on their environment. Or in other words, we can say that these are environment-dependent cancers. Now, such tumors that are dependent on their environment. One of their remarkable example is teratocarcinoma, and it is a tumor in germ cells or stem cells. Now, these cancers, the teratocarcinomas, terato also links this cancer to development. Now, these teratocarcinomas, these are malignant growth of cells that resemble the inner cell mass of mammalian blastocysts, and thus they can kill the organism. They are what you can say lethal. Now, if a teratocarcinoma cell, which is a cancer cell, it is placed in inner cell mass of a mouse blastocyst, it will integrate into the blastocyst, lose its malignancy and divide normally. But we are saying that teratocarcinoma was a malignant growth that resembled mammalian blastocyst. It was not mammalian blastocyst, but it was a cancerous growth resembling mammalian blastocyst. And when, as it was similar to blastocysts, researchers took some of the cells from teratocarcinoma and they placed these cells in normal uh, blastocyst of mouse. But what was quite astonishing to observe was that in this case, when cancer cells were placed on the inner cell mass of normal blastocyst, those cancer cells lost their malignancy. Means their malignancy was dependent on the environment they were in. They are what you can say context dependent tumors. Now, its cellular progeny can become part of numerous embryonic organs. Those cells that we integrated into the blastocysts, that we placed the inner cell mass of blastocyst, when that blastocyst is going to complete its embryonic development, those cells that were once what you can say malignant, those cells are now going to become the part of numerous embryonic organs. And now these cells that were malignant, but they lost their malignancy because they were in a different environment. When these cells are going to become the part of numerous embryonic organs, and let's say that they become part of the germline. Now, this germline it is going to give rise to eggs and sperms. So, when these tumor cells these are going to be part of germline. So the sperms or egg cells that will be formed from these tumor cells, will they transmit the tumor genome to next generation? Well, that is the question, and that is a possibility. Thus, whether the cell becomes a tumor or becomes part of embryo, it can depend upon its surrounding cells. It is possible that the stem cell environment suppresses the tumor formation by its secretion of inhibitors of paracrine pathways. Or in other words, what we can say that during the early embryonic stages, uh, when we implanted that malignant keratocarcinoma cells into the mouse blastocyst, they had what you can say a stem cell environment, and that stem cell environment suppressed the formation of this tumor by the various secretions, various inhibitors of this tumor formation, and they involved paracrine pathways. Now, for instance, let's say that many tumor cells, such as the melanomas, they secrete paracrine factor nodal. Now, what is going to happen? This paracrine factor nodal that is secreted by melanomas, this aids in their proliferation and also helps them supply with blood vessels. Now, this nodal paracrine factor that is released by melanomas. What we do if we place these melanomas in an environment of embryonic stem cells? We take these melanoma cells that are secreting this paracrine factor, and we place these melanoma cells in an embryonic stem cell environment which secrete nodal inhibitors. Now, what is going to happen is that the paracrine factor nodal that has been released by melanomas to help them proliferate, that is going to be suppressed by this nodal inhibitor that is secreted by the embryonic stem cells. So, aggressive melanoma cells, they become normal pigmented cells when they are made part of embryonic stem cells. And remarkably, such malignant melanoma cells, when they're transplanted into early chick embryo, what you can say, when they're placed in early chick embryo, these malignant melanoma cells, they downregulate their nodal expression. In other words, we can say that not there are this is not only the case that the nodal inhibitors from embryonic stem cells, they are going to, what you can say, compensate for the nodal paracrine factor from melanomas, but in addition to that, the expression of this nodal paracrine factor, it is going to get down regulated. And in other words, we can say that both of the things are happen. The first thing, that the nodal paracrine factor is being, what you can say, inhibited by the nodal inhibitors in the embryonic stem cell environment, and the early chick embryo, or the, what you can say, the stem cell environment, that is causing these malignant melanoma cells to downregulate their expression of this nodal paracrine factor, and thus these malignant carcinoma cells, malignant melanoma cells, they are going to migrate as non-malignant cells along the neural cluster pathways. So this is how the context dependent tumors, uh, they are caused, and they are, what you can say, regressed or repressed. Now let's take a look at this grammatically. So here we have, what you can say, aggressive metastatic human melanoma cell, and they are injected into a 2-day chick embryo. What you can say in simple words, they are getting injected into a stem cell environment. Now they are going to form what is going to happen during the normal development. They are going to form the normal migratory chains, and they are going to follow the neural crust migration routes to integrate into the facial cartilage. So they are these metastatic melanoma cells. They lost their malignancy and they became normal cells, and they become the part of our normal facial cartilage, or the normal facial cartilage of chick, here to say, as a result of being implanted into a stem cell environment that regressed their growth, that repressed these melanomas from being metastatic in a number of ways. Now, after studying that cancer can be influenced by the environment, and cancer cells influence their environment in order to be able to proliferate, we are going to go towards the next topic, that is cancer because of defects in the cell-cell communication. Now, in many cases, the tissue interactions, they are required to prevent cells from dividing. Uh, What we can say, during developmental process, the interaction of certain tissues with other tissues or cells, that leads to the, what you can say, prevention of division in the other cells. Now, if this interaction, it is somehow, what you can say, it becomes abnormal or it is altered, it can lead to cancer. How it can lead to cancer? Because the cells that was supposed to be prevented from dividing, they are not going to be prevented from dividing, and thus they are going to get uh, be able to divide, and form cancerous growths. So cancer can be caused by the miscommunication between cells. So we can say that tumors can arise through defects in tissue architecture, and the threading of a cell are critical in determining malignancy. And studies have shown that tumors can be caused by altering structure of a tissue, and these tumors can be suppressed when restoring appropriate tissue environment. Again, the processes that are important in the developmental uh, changes, this tissue architecture, this tissue environment, they are also influencing what you can say carcinogenesis or oncogenesis and metastasis. So, in particular, 80% of human tumors are from epithelial cells. And now these cells, they do not always appear to be the site of cancer causing lesions. Rather, though the cancer is being of these epithelial cells, rather the epithelial cell cancers are often caused by defects in the mesenchymal stromal cells. So, who are getting cancer? Epithelial cells. But what is causing cancer? That is the defect in mesenchymal stromal cells. And these mesenchymal stromal cells are the ones that surround and sustain epithelia. Researchers, when they recombined normal and carcinogen treated epithelia and mesenchyme in rat mammary glands, the tumorous outgrowth of mammary epithelial cells occurred not in the carcinogen treated epithelia but only in the epithelia that was placed in combination with mammary mesenchyme that has been exposed to carcinogen. So, what we are saying in simple words that though the cancer was occurring in epithelial cells, it was not caused by epithelial cell or the changes in epithelial cell, but rather it was because the carcinogens altered the mesenchymal cells, and those mesenchymal cells they made some sort of miscommunication with those epithelial cells, and that miscommunication between these mesenchymal cells and the epithelial cells that led to the cancer of epithelial cells. That's the carcinogen that caused defects in mesenchymal stroma of mammary gland, and uh, apparently the treated mammary stroma could no longer provide the epithelial cells the instruction to form normal structures. This mesenchymal stroma, it was supposed to provide the normal instructions to epithelial cells for normal growth of the structures, but when it was treated with carcinogen altered the mesenchymal stromal cells, and that led to a miscommunication between these mesenchymal stromal cells and the epithelial cells, and it led to the development of cancer, or what you can say, the growth of cancer in epithelial cells. So, this is another way in which developmental biology is relevant to, or is related to, is highly related to oncology, or in other words, what we can say that the changes that were normally associated with developmental process, these changes are also associated with cancer proliferation, carcinogenesis, and metastasis. Now, this is what we were talking about, how the stroma cells or how the mesenchymal cells they are going to influence epithelium cells. Let's say that here is a section of mammary gland tissue, and mammary gland contains both these epithelial cells or epithelium and the mesenchymal cells. Now, what we can do is that we can separate these two types of cells, and we can do either of the three things. The very first thing that we can do is that we can expose epithelium only to some carcinogen and then place it over normal stroma. The other thing that we can do is that we can use these stroma cells and we can expose these stroma cells or these mesenchymal cells to carcinogen or mutagens, and then we can place these stroma cells that were exposed to carcinogen or mutagen, in other words, alongside normal epithelial cells. Or what we can do is that we can uh, expose both of these cells, the epithelial cells and stroma cells, to the mutagen or carcinogen. This is what is going to be uh, happening schematically when we are exposing the stroma cells, or what you can say, this control vehicle to these stroma cells, that is going to influence the epithelial cells. And depending on whether we exposed the stroma cell to carcinogen or the normal vehicle, it is going to get it is going to get going to influence the epithelial cells to either develop the cancer or not develop the cancer. And here are what the results look like. Let's say that we exposed epithelial cells with a normal vehicle. However, we uh, exposed the mesenchymal cell with this what you can say N-methyl nitro L-methyl nitrosourea, which is a mutagen. So when epithelial cells were exposed to VEH, or what you can say that control vehicle, and stromal cells they were exposed to this mutagen, there were high rates of cancer. Similarly, when the epithelial cells were exposed to mutagen and the stromal cells were also exposed to mutagen, in this case there were also high cases of cancer. However, when the epithelial cells they were exposed to and epithelial cells were exposed to mutagen and the stromal cells they were not exposed to mutagen, what happened is that there was no cancer present in the epithelial cells. And when both of these cells were exposed to the control vehicle, uh, there was no cancer as well. This means that cancer is only going to be get caused when the stroma cells or the mesenchymal cells they are going to get exposed to mutagen here in this case and here in this case as well. However, if the what you can say carcinogenesis or the development of cancer it was related to epithelial cells, then cancer would have been here in this case as well when epithelial cells were exposed to mutagen. So we can say that in positive control there were high cases of high rates of cancer, and in negative control there were no cases of cancer at all. So this is how we came to realize that the mesenchymal cells, their miscommunication with epithelial cells, it can lead to the formation of cancerous growths in epithelial cells. Now let's move on to our next topic, which are the defects in paracrine pathways. So by now we have studied that there can be context dependent tumors, or the tumors can also be caused by that miscommunication between cells or the tissue and cells, etc. Now we are going to study that cancers can also be caused by the defects in the paracrine pathways. So the tumors can occur by disruption of paracrine signaling between cells. And several key signaling pathways such as a hedgehog, notch, WNT, BMP, TGF, beta, activin, these paracrine pathways they are involved in most processes that are essential to normal development. Now again, whatever was we thought that it was only associated or it was only related to normal proper development, it is going to somehow able to cause cancer when some kind of abnormal changes occur. It is becoming increasingly clear with more and more research being done into this topic that these paracrine pathways they can have a crucial role in tumorigenesis, or what you can say similar word to carcinogenesis or oncogenesis, uh, growth of cancer. So when these pathways that were supposed to function only during embryonic life, when these paracrine pathways they are reactivated in adult tissue through what you can say sporadic mutations or other mechanisms, this can become the cause of cancer. Above, we saw that the nodal secretion by melanoma cells, the communication subtopic. So, nodal secretion by melanoma cells, they what you can say influence the surrounding tissue so that surrounding tissue supports that cancer or promotes that cancer. Now, these findings demonstrate the importance of stromal cells, which are which we just have studied. Now, many tumors, for instance, uh, they secrete a paracrine factor which is sonic hedgehog or SHH. Now, this paracrine factor that is secreted by tumor cells, it can act in two ways. First of all, this can act as an autocrine, what you can say, uh, signal molecule, or it can influence the cell that is releasing uh, uh, but you can say that this paracrine factor SSHA, it can act in an autocrine way or it can influence the cell that is producing it. In this case, the stimulate it can stimulate the cell that produce it to grow. This autocrine SSH is normally required for the maintenance of what you can say the cerebral granule neuron progenitor cells and also the hematopoietic stem cells or the cells that are what you can say in simpler words become the precursors of blood cells. So this SSH when acting in paracrine fashion it can have these important roles. However, the SSH pathway inhibitors they can reverse certain what you can say medulloblastomas and leukemias. These are cancers. So when we observed that when we apply this SSH pathway inhibitors, it causes the reversal in certain cancers. It means that this paracrine factor is somehow affecting the development of cancer. Or we can say again that in this way, the normal developmental process, when they are altered or when they, you can say, become abnormal, those developmental process are going to lead to cancer. Now, an autocrine requirement for the hedgehog has to be reported for uh, what you can say small cell lung carcinoma. It is another another kind of cancer, and the pancreatic adenocarcinoma, the prostate cancer, and the breast cancer, and colon cancer, and liver cancer. So all of these cancers they involve some kind of uh, involvement of this SSH in an endocrine in an autocrine fashion. So this means that there is some kind of abnormality in this paracrine pathway. Now the second function that is of this SSH is that this SSH produced by tumor cells, it may not act on tumor cells themselves, but on the stromal cells, and thus, or what you can say on the mesenchymal cells. There's similar thing that we studied earlier, the mesenchymal cells, when they were exposed to uh, some carcinogen or some mutagen, there was miscommunication between mesenchymal cells and between the epithelial cells, causing cancer in epithelial cells. So this can be the possible cause that the stromal cells they are having some disruptions in their paracrine pathway, in their SSH pathway, and that is leading to the development of cancer. So these stromal cells they can produce factors such as insulin-like growth factor IGF, and this insulin-like growth factor it can support the tumor cells. Let me simplify a bit. Well, the mesenchymal cells they are producing SSH, and this SSH is functioning in autocrine fashion, or which means in simple words, the stromal cells are producing this SSH, and this SSH is in turn acting on same stromal cell that are producing it. Now, how it is influencing that stromal cell that is producing it? It is causing that stromal cell to produce factors such as insulin like growth factor or IGF. And now this insulin like growth factor, it is going to support the tumor cells. If the SSH pathway is blocked, the tumor regresses. For example, cyclopamine, which is a teratogen, and it blocks SSH signaling, it can prevent certain of these tumors from growing. Isn't it astonishing? The same chemical that during embryonic development is causing teratogenesis or abnormal development, the same chemical when in the adult life, it when it is used in adult life, when there is no that developmental process that are occurring in the embryonic development. So now this teratogen it can block the growth of cancers. It means that cancers are somehow related to developmental abnormalities as well, or we can say that cancers can be studied as a disease of development. Thus, the same chemicals that can cause teratogenesis by blocking a pathway in embryonic development, they can be useful in blocking the activation of cancer stem cells. Now, this cyclopamine and other antagonist of SSH pathway, they can cause malformation in embryos, but they appear to be useful in preventing the generation and proliferation of medulloblastoma stem cells. Thus, in embryonic life, they are going to harm, but in post embryonic life, in adult life, when there is no going to be any uh, necessity for developmental changes that had occurred in embryonic life, it can lead to what you can say the reversal of cancer. Even the classic teratogen thalidomide, thalidomide, this teratogen is being rehabilitated, or what you can say is being thought again in as a potential, what you can say, anti-cancer drug. So by now, in simple words, we have seen that the normal processes that were occurring in embryonic development, when they for some reasons occur again during adult life, it can lead to cancer. And so the teratogens that were disrupting, that were able to disrupt that process during embryonic life, those teratogens can also be used in adult life to suppress the same process, and thus in adult life it can suppress the process when it is not required and it is cancer causing. The fourth subtopic that we're going to study today, the cancer stem cell hypothesis, or the involvement of cancer and stem cells. I said in opposite way, or the involvement of stem cells in cancer. Now, another aspect of viewing cancer that a disease of development is the properties of tumors may emerge because of a population of cells that are analogous to adult stem cells. So, the idea that cancers had stem cells was one of the first links that connected this cancer research and developmental biology. It was demonstrated by researchers, or it was reported by researchers in 1971, that malignant tissue, like normal tissue, it maintains itself by proliferation and differentiation of its stem cells. So, just like normal stem cells that are going to help maintain the normal tissue, cancer cell is also having stem cells of its own that are going to maintain that cancer. And in the same year, it was demonstrated by researchers that the presence of stem cells were uh, the same year, it was reported that there might be the stem cells that cause the maintenance of cancers, and in the same year it was proven that in rat carcinomas such stem cells are present. And the similarity between normal stem cells and these cancer stem cells, it was highlighted when the linear tracing revealed, it's a technique we're not going to discuss in detail at all. So this, when linear tracing was used, it revealed that the stem cells of intestinal adenomas are precursors of intestinal cancer. They are LGR5 positive, and they have some relationship to the Paneth cells, as do the normal intestinal stem cells. Or in simpler words, in non-technical terms, we can say both the normal intestinal stem cells and the stem cells that were part of precancerous growth both had same relationship to the Paneth cells, and thus they were, we can say in simple words, they were related to. And in numerous cancer growths such as glioblastomas, which are the most common brain tumors, and in prostate cancer and in melanomas, let me highlight it, and in myeloid leukemias, a rapidly dividing cancer stem cell population, or CSC, cancer stem cell population, it gives rise to more cancer stem cells and to the population of relatively slow dividing differentiated cells. Thus, cancers also involve stem cells. Now, these cancer stem cells, they can renew as well as they can generate the non-stem cell population of tumor. So these stem cells, these cancer stem cells, they are going to what you can say promote cancer in two ways. First of all, they are going to self-renew themselves, thus producing more and more of these cancer stem cells. And also, these cancer stem cells are going to give rise to the non-stem cell population that is going to form that tumor. So when tumors cells that were transplanted from one animal to another, only the cancer stem cells they gave rise to the heterogeneous tumor. It means that it might be possible that those non-stem cell populations in a tumor they were not responsible for the tumor growth, but rather only the stem cells, the cancer stem cells that were part of tumors, they were giving rise to the tumors, the heterogeneous tumor. Now, the origin of these cancer stem cells it remains uncertain, and it may be different from different. It may be different for different type of tumors, and most researchers feel that these cancer stem cells they come from either a normal adult cell or a progenitor cell that is what you might think get mutated or get changed in some abnormal way. While the tumor is forming, these stem cells, these cancer stem cells, they produce more cancer stem cells as well as the bulk of more differentiated tumor cells, as we have seen. And remarkably, it appears that CSCs or the cancer stem cells of aggressive glioblastomas or brain cancer that we just studied above there. So these cancer stem cells, they not only make immature glia-like cells or glioblasts that are part of cancer, but they can also make blood vessels and endothelial cells. And this is a property of stem cells that they are multipotent, they can produce various structures, and hence it is occurring in this case, in the case of aggressive glioblastoma cancer stem cell, that they are not only forming the glia-like cells or glioblasts, they are also forming blood vessel endothelial cells. In this way, cancer or tumor can create its own vasculature or it can create its own blood supply. So this was what you can say all about cancer as a disease of development, or this is how we can say teratogenesis or developmental abnormalities are related to oncogenesis, or what you can say carcinogenesis, or what you can say tumorigenesis, or what you can say in the metastasis of cancer. Now proceeding further and talking a little bit about the development therapies for cancer, as we know that cancer is associated with developmental biology. So treating the developmental disorders can treat cancer. So we can say that cancer is not a result of a single cell gone bad, but it is kind of a relationship between cells that is gone awry, or that is what you can say gone unhealthy. Now, cancers are often disease of development signaling, or several type of cancer stem cells, or sorry, the several type of cancer cells can be normalized when they're placed back in embryos that express certain paracrine or their inhibitors, as we have seen earlier. Now, this developmental view of cancer it allows us to explore new ways in which we can treat cancer. And one of such modes of treatment is differentiation therapy. And this differentiation therapy is not something new. It was considered as long as 30 years ago, but at that time it was not feasible. In 1978, researchers noted that cancer cells were in many ways reversions to embryonic cells, as we have been seeing in this lecture. And thus, these researchers hypothesized that cancer cells should revert to normalcy if they were made to differentiate. Thus, in 1978, the same year that it was noted by researchers, it was discovered that certain leukemias they can be controlled by making leukemia cell differentiate rather than proliferate. So, the one thing that about cancer that we should know is that cancer cells are poorly differentiated. Cancerous mass or tumor is poorly mass of poorly differentiated cells. They are just doing one thing, that is to divide, 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 divide. So what if we somehow make them switch from divide, divide, divide mode or proliferate mode to differentiate mode, so that there is no more proliferation and thus the cancer reverses. And one of these leukemias, which is APL or acute promyelocytic leukemia, it is caused by somatic recombination creating a new transcription factor, and one of the whose subunit is retinoic acid receptor. So this leukemia, it is caused by what? It is caused by uh, what you can say some problem that is going to give rise to this new transcription factor, And this new transcription factor has a subunit which is retinoic acid receptor. This receptor, even in the absence of retinoic acid, it binds to the retinoic acid binding sites in DNA. What was actually supposed to happen is that this receptor was supposed to bind retinoic acid in DNA to DNA, but in such kind of leukemias, what is happening instead, what is going on instead, is that despite retinoic acid being absent, these receptors are binding to DNA. Now, when this receptor that is not bound to retinoic acid, when this receptor is going to bind to DNA, what it is going to do that it is going to repress the RA or retinoic acid responsive genes. So now, and these retinoic acid responsive genes, they are going to get repressed, or they are going to get uh, down regulated. It creates a large condensed chromatin structure, and this large condensed chromatin structure, this is what is going to cause the problems for the cancer. An expression of this new transcription factor in neutrophil progenitors, it causes the cell to become malignant. And the treatment of APL patients with all transretinoic acid, it results in remission in more than 90% of the cases, because the additional retinoic acid is able to affect the differentiation of leukemia cells into normal neutrophils. So what was happening is that, here let's revise it. This APL was being caused when a certain transcription factor formed in the cells. This transcription factor has a subunit that was what you can say a retinoic acid receptor. But instead instead of what happens normally, that retinoic acid binds to this receptor and then this receptor binds to DNA. This retinoic acid receptor, it was not bound to retinoic acid, and even then it was binding to DNA. So when it was binding to DNA without the presence of retinoic acid, it was going to cause the repression of RA responsive genes. It was taking their place. It was the taking place of normal retinoic acid receptors or normal retinoic acid bound receptors. So when retinoic acid was given to these, all trans retinoic acid was given to these patients of APL, in 90%, more than 90% cases, there was remission in the cancer, because this RA, this retinoic acid, was able to affect the differentiation of leukemia cells in normal neutrophils. Now, recently, what we have been trying is that are trying to use microRNAs in differentiation therapies, and in many tumors that are what you can say, uh, in many tumors there are specific microRNAs that are downregulated. We know that microRNAs, they have a very crucial role in various kind of regulations of gene expression. So when these microRNAs, these are downregulated, it means that the gene regulation is going to get affected, and that effect on gene regulation, that is going to cause cancer. So what we can do is that these downregulated microRNAs, which are usually tumor suppressors and prevent the DNA changes, prevent changes in DNA methylation. What we can do is that we can use these microRNAs, for example, in 2009, such as such as have shown that microRNA microRNA 206, which is normally present in skeletal muscle cells, and it is downregulated in tumors. If we add this microRNA 206 to muscle tumor cells, it restores their differentiated phenotype and it blocks cancer formation. Thus, in this way, we can treat cancer by studying developmental biology. And this is how this lecture comes to a conclusion. I hope that you found this lecture of appreciable quality, as well as of what you can say, you have found in this lecture the content that you desire to find. To find. So if it is so, then please Do like this video and do share your opinions with me in the comment section. And do not forget to subscribe to this channel and turn the notifications on. See you in the next lecture. Thank you so much for watching.